Hi, everyone. I'm Mr. Red, a red blood cell in my friend Long's body. He told me he really enjoyed his biology class and invited me to give an introduction of it. So here I am. Welcome to the cellular ballet, witnessing the dance of division in BIOL 110. You may not see us, but right now, trillions of my cell brothers like me are busy working, repairing, and my personal favorite, dividing, even if I can't. Today, I'm going to take you backstage and show you how they pull off this beautiful, organized chaos called mitosis. In order to avoid covering the content, please forgive me for my disappearance. Thank you very much. First big question, why do my cell brothers even bother dividing? Can't they just chill? Unfortunately, no. Life would be a disaster. For development, you human beings started as one tiny fertilized egg. With round after round of division, you leveled up into a full human. For growth, you didn't just stretch your baby cells like pizza dough, you made more of us. For repair, when you scrape your knee or get sunburned, damaged cells retire and new ones take their place. No cell division means no growth, no healing, no you. So yeah, this is not a side quest. This is the main storyline. Today's episode is about mitosis, the kind of division done by your somatic cells, skin cells, gut cells, blood cells, all the everyday heroes. Their job in mitosis is simple but strict. Start as one diploid cell, divide to make two diploid daughter cells, each with a perfect copy of the DNA from the parent cell. It's like photocopying an important 1,000-page book twice. No missing pages, no extra chapters about how to become a TikTok star. Don't mix this up with meiosis. That's for making sperm and eggs, where they have the chromosome number. Totally different show. Before the show, their DNA hangs out as relaxed, messy chromatin. Comfy, but not stage ready. Right before division, they replicate their DNA in S phase, so every chromosome has two copies. Pack it up neatly into chromosomes you can actually see. Each one turns into an X shape. Two identical sister chromatids held together at a centromere, with protein clips called cohesins. Later on, those clips will get cut, and the twins will dramatically run to opposite sides of the cell. No spoilers. Okay, a little spoiler. Fun fact, mitosis is actually the shortest part of their lives. Most of the time, they're in interphase, doing normal cell things. In G1, they grow, make proteins, pay taxes. Just kidding, mostly metabolism. In S, they replicate DNA, so every chromosome has two sisters. In G2, they finally grow and quality control, build the gear for division. Then comes the M phase, where they split the nucleus and chromosomes, and finally split the cytoplasm, actually forming two cells. Checkpoints along the way make sure everything's safe. When those checkpoints break and cells divide without control, that's when we start talking about cancer. So yes, this choreography needs to be perfect. Lights down, spotlight on, mitosis begins. In prophase, my cell brothers' chromatins condense into visible chromosomes. The dancers put on their costumes. The centrosomes move to opposite sides of the cell. The mitotic spindle, made of microtubules, starts to form like strings across the stage. In prometaphase, the nuclear envelope breaks into pieces. Bye-bye barrier. Microtubules reach in and attach to chromosomes at kinetochores, special protein handles on each centromere. Some of my brothers told me that it feels like being hooked up to dozens of tiny fishing lines all at once. Not super comfortable, but scientifically very important. Next up, the Instagram moment of mitosis. In metaphase, every chromosome lines up in the middle of the cell on the metaphase plate. Each one is connected to microtubules from both poles, so the pulling force is perfectly balanced. If you've seen one textbook pic of mitosis, it was probably this stage. Everything looks neat, symmetric, totally under control. Then, chaos, but organized chaos. In anaphase, the cohesins are cleaved. Sister chromatids officially become daughter chromosomes. Spindle microtubules shorten and pull them toward opposite poles. It's like someone yelling, "Roommates, you're moving out!" And suddenly everyone sprints to a different side of the apartment. I'm back! Finally, we calm down. In telophase, chromosomes arrive at the poles and start to decondense back into chromatin. 2 new nuclear envelopes form, one for each future daughter cell. The spindle disassembles, the stage crew goes home. In cytokinesis, in my animal cell brothers, a cleavage furrow forms. The membrane pinches in like tightening a drawstring bag. In plant cells, instead of pinching, a cell plate grows in the middle and becomes a new cell wall. As a result, 2 daughter cells, genetically identical and ready to start the cycle again. They bow, the curtain closes, and then they do it all again in another tissue. Okay, theory is nice, but BIOL 110 made you do something cooler. You actually watched cells, like my brothers, dividing in real life. In Lab 3, you will get the chance to see this incredible process for yourselves. In this lab, you will prepare a specimen from garlic root tip and observe the different stages of mitosis using a compound light microscope. Why garlic root tips? Because the root tip's apical meristem is a mitosis hotspot. It's basically Broadway for cell division. Your protocol had 4 main parts. First, you cut about 1 centimeter from the root tip, fixed it in a fixative to preserve the cells and the structures, and then soaked it in 1 molar hydrochloric acid to loosen the cell walls. Next, you stained the tissue with 0.8% gentian violet so the chromosomes would bind the dye and become bright purple under the microscope. Then you placed the stained root tip on a slide, added a cover slip, and pressed gently straight down to spread the cells into a thin single layer without twisting. Finally, you observed the slide with a compound light microscope, starting at low power to find the right region, and then moving to high power to identify cells in different stages. For you, that was a lab practical, but for us cells, it was basically a surprise photo shoot. However, for science, we cooperated. This is the masterpiece captured by Long, and he very firmly told me that his results must be shown to everyone. What you are looking at is my entire extended brothers on a garlic root tip, frozen in different moments of the mitotic dance. In the middle, you see the crowded stage, and around it, Long has zoomed in on individual performers in interphase, prophase, metaphase, anaphase, and telophase. So you can actually point to real cells and say, this one is getting ready, that one is lining up chromosomes, and that one is pulling them apart. By studying this slide, you are no longer just memorizing a textbook picture. You are watching real cells, like me, do our everyday job of dividing, which definitely makes Long's result worth showing off. So why should you care about our little mitotic ballet beyond the quiz? In cancer biology, tumors form when cells ignore the normal cell cycle checkpoints and keep dividing when they should stop. And many chemotherapy drugs target stages of mitosis in order to slow those rogue cells down. In developmental biology, the same basic steps you saw in garlic roots are used to build hearts, brains, leaves, and flowers. In regeneration and stem cell research, understanding and controlling cell division is essential for repairing damaged tissues. That purple field of cells that you counted under the microscope follows the same logic that doctors and researchers use when they examine dividing cells in a biopsy from a patient. What I love about BIOL 110 is that it doesn't stop at, here are the names of the stages, please memorize them. Instead, you learned the concepts of the cell cycle and mitosis, practiced real lab skills, not just pipetting, but also patience and troubleshooting, and connected what you saw on your slide to big ideas about growth, disease, and life itself. From my side of the membrane, that's when students stop being people who read textbooks and start becoming scientists. So, on behalf of Long and cells everywhere, thank you for watching our performance and not just skipping straight to the exam review sheet. Long also asked me to remember to express his huge thanks to his instructor Ju-Hyun Lee and TAs, his classmates and lab partners, and his families and the DKU community for supporting this journey. Next time you get a paper cut, grow a little taller, or just blink. Remember, somewhere inside you, Millions of my brothers are quietly performing the same mitotic choreography you just learned. I'm Mr. Red. Welcome to join our Bio 110 family. Signing off. Stay curious, keep observing, and please handle your slides gently. We're on them.